Welcome to Inert Chemistry. In today's video lecture, we are starting discussing lactic acid fermentation or lactic acid production at industrial lab. Here is the introduction about lactic acid. Lactic acid it is a very important material. It is used in food, pharmaceuticals, and textile industry. Lactic acid it's an organic acid. Its molecular formula is CH3 CHOHC OOH. Here you know that there are two forms of lactic acid, L-lactic acid and D-isomer of lactic acid. L-lactic acid, the liver rotatory form, it is obtained by biological fermentation of sucrose, and D-lactic acid, also called dextro-rotatory form, it is present normally in blood and muscle tissues as a product of anaerobic metabolism of glucose and glycogen. It is having various uses in our daily life. Its application include food preservation, pharmaceutical and chemical industries. Its water retaining capacity makes it useful as a moisturizer in cosmetic formulations. Due to presence of hydroxyl and carboxyl group in its structure, it can be converted into useful chemicals like esters, biosolvents, etc. What is the raw material for the formation of lactic acid? The cheap raw material is required. You will have to choose the cheap raw material for the process of farming of this product. It should have the ability to produce high yield. It should be negligible or no formation of byproduct. These are the various characteristics that must be present in the raw material. The first one, it should be cheap. It should have the ability to produce high yield. It should be negligible or no formation of byproducts, and it should have the ability to take less contamination so that not much pre-treatment is required. Following are some of the cheap raw materials that are used, like starchy material that includes your corn, potato, rice, etc. Cellulosic material like wood and cellulose. Agricultural residues like corn cob, cassava, bagasse, wheat, molasses, etc. Whey contains lactose, protein, fat, and mineral salts as it is a product of dairy industry. The microorganism. The microorganisms that are capable of production of lactic acid they include bacteria, fungi, and yeast. So let's discuss about this. One of the key reasons for the use of lactic acid bacteria in industry, lactic acid bacteria are actually those type of bacteria that uh, are having the ability of fermentation of lactic acid. So one of the key reasons for use of lactic acid bacteria in industry is because it does not have any adverse health effects. Lactic acid producing bacteria are classified into four main categories. Here you see lactic acid bacteria that are called LAB, E. coli, and similarly there are some bacillus strains are also present, and Corynebacterium, Glutamicum, these are also very important. Here two types of microorganisms are actually used, homofermentive and heterofermentive. Homofermentive, they convert glucose excessively into lactic acid, while heterofermentive, they produce less yield of lactic acid due to the formation of various byproducts. Fungi. Advantage of fungal fermentation over the bacterial fermentation is that the fungal fermentation it is low cost downstream process. Low nutrients are required for fungal fermentation. Formation of fungal biomass, which is an important byproduct. Fungal fermentation use chemically defined medium, so the purification of products is very simple. This is major advantage in food industry. The organisms of the genus Rhizopus are demanded as the best fungal sources for lactic acid production by fungal fermentation. Yeast, Saccharomyces cerevisiae, it is also commonly used. The major advantage of using yeast is that Their tolerance against low pH means this yeast can tolerate the low pH conditions. It is having ability to grow in minimal mineral mediums, and various species of yeast are commonly used for the production of high yield of lactic acid. What are the fermentation conditions? The first condition of temperature. The temperature must be around 45 degrees centigrade or comparatively higher temperature. The lactic acid fermentation is carried out at comparatively higher temperature, that is 45 degrees centigrade. What is the pH conditions? The fermentation proceeds best when pH is on the acidic side. Sugar concentration. The sugar concentration in masses is normally adjusted at a concentration of 5 to 20%. Oxygen. The bacteria used to produce lactic acid industrially are usually micro aerophilic or anaerobic in nature. This is the you can say that this is the processes of your lactic acid fermentation. First of all, you will have to take sugar and you will have to perform its hydrolysis. Due to hydrolysis, it is converted into this complex sugar is converted into simple monosaccharides or simple glucose. Then you will have to perform its glycolysis. Due to glycolysis, the end product pyruvate is produced. You will have to perform fermentation of pyruvate that gives you various fermentation approaches are as follows. It may be batch fermentation, fed batch, and continuous fermentation. Various other processes for the production of lactic acid by chemical synthesis include oxidation of propylene glycol, nitric acid oxidation of propylene, etc. But none of these chemical synthesis processes are economically and technically feasible. So first of all, we will see batch fermentation. In batch fermentation, all the required materials such as carbon source, nitrogen source, and other components they are added prior to beginning of fermentation process. Batch fermentation is further classified as solid state fermentation and separate hydrolysis and fermentation process. Fed batch fermentation, it is the second type or second approach of your fermentation. In fed batch fermentation, all the required material like carbon, nitrogen, and other required compound they're added during fermentation process at regular intervals of time without the removal of fermentation broth. Continuous fermentation. Continuous fermentation involves the addition of fresh medium to the fermenter while withdrawing the already existing broth at the same rate. It is having various advantages: prevention of end product inhibition. Sometimes your end product it inhibits the process, but in this way you will have to prevent the end product inhibition. It is having less frequency to shut down, less decrease in productivity during lag phase, and there are also various advantages. Here is the summary of the various processes that occur during the fermentation. Product recovery by precipitating techniques. There are different techniques that are performed for the recovery of your process. Separation by precipitation. It is the most conventional primary purification step in lactic acid production by fermentation. It includes the various steps. The first one is neutralization. Excess calcium carbonate or calcium hydroxide, it is added to the fermenter to neutralize the acid produced, maintaining the pH around 5 to 6 and producing a calcium salt of acid that is called calcium lactate. If the lactic acid are not neutralized, then LAB means lactic acid bacteria. They would not be able to tolerate the high acidity develop and the fermentation would not continue to completion. The second step is acidification and filtration. Sulfuric acid is added to precipitate the calcium sulfate or gypsum, which is filtered. The third step is centrifugation. It removes the biomass impurities. And the fourth step is evaporation. The filtrate containing free organic acid is evaporated to obtain the pure lactic acid. Here you say that during the fermentation, first of all, what will happen that your sugars are converted to lactic acid. During the recovery, first of all, we will have to perform the neutralization step. What happens in the neutralization step? We will add calcium hydroxide here. This calcium hydroxide, it neutralizes your lactic acid by forming calcium lactate and water. It is a neutralization step. After that, you will have to add some acid in it, like sulfuric acid. This sulfuric acid, the fermentation broth is treated with sulfuric acid in order to precipitate your calcium sulfate, or also called gypsum. When you add sulfuric acid, your calcium sulfate or gypsum, it is you can say it get precipitated, form precipitations, and there are some disadvantages of this technique. The second technique, it is the product recovery by solvent extraction process. Liquid-liquid extraction, also known as solvent extraction, it's the method to separate compounds or metal complexes based on their relative solubilities in two different immiscible liquids. Usually one of these is polar and the other is non-polar. Solvents are selected to present higher affinity to lactic acid and little or no affinity to water. So this technique, liquid-liquid extraction, is also used, and during which various specific types of solvents are used that show affinity with lactic acid and do not show affinity with your polar water. Product recovery by membranes. Membrane separation process that are based on the transfer of solute through a semi-permeable physical barrier that separate the two phases, restricting the transport of components of one phase to another. This is also very common. Here we quickly review. First of all, you will have to take the raw material, and then you will have to take microorganism, and you have to perform fermentation. There are three major types: either batch fermentation, fed, or continuous fermentation. After for your recovery process, you will have to perform the neutralization first of all, then acidification and filtration, then you will have to centrifuge your material, then evaporate. After that, you will have to dry your product, and finally packaging and then shipping. Thanks for watching this video.